Hello, everybody. Today we have a double header from the University of Dundee. So first, please welcome Dr. Charlotte Crowe, a postdoctoral researcher in Dr. Alessio Cioli's lab at the Center for Targeted Protein Degradation. Originally from Strasbourg, France, Charlotte moved to Scotland in 2015 to study chemistry at the University of St. Andrews. She completed her PhD on small molecule degraders and E3 ligases at Dundee and joined the CTPD Light project in 2025. And she's been working with the Michael J. Fox Foundation now to develop new therapies in Parkinson's diseases. So without further ado, please, Charlotte.
Wonderful. Thank you so much for the introduction. I'm just going to start sharing my screen now, which you should now be able to see. Yeah. Thank you for the introduction and for the kind invitation to give a talk. I was really excited when I received the email and excited to show you about some of the work that we've been doing in the Center for Targeted Protein Degradation. So a quick introduction on the centre is we're located in Dundee, which is on the east coast of Scotland. And back when I started my PhD here in 2020, we were located in the main School of Life Sciences in Dundee. But then back in 2023, we packed up all of our boxes and lab equipment and moved ourselves across the road, really just a 2-minute walk away to our new Centre for Targeted Protein Degradation, which was established in January 2023 by my supervisor and our director, Alessio Cioli, who became really good at opening bottles of champagne that year, given how many inauguration events we held to celebrate. And our main mission in the Center for Targeted Protein Degradation is to develop small molecule degraders which hijack the ubiquitin proteasome system in order to degrade disease-causing proteins. And as part of that, what we need to do is understand the underlying mechanisms of this degrader pathway in order to develop and optimize better degraders. So in targeted protein degradation, what we're doing is we're introducing a small molecule degrader into the system. And so for the purpose of this talk, I'm just going to be talking about Protacs, which are proteolysis targeting chimeras. And these are typically heterobifunctional in structure. So they're composed of a substrate recruitment part, an E3 ligase recruitment part, and these two moieties are bound by a linker region. And so if we think about the degrader pathway, what we need to do is allow the Protac to first enter the cell. And then once it's arrived in the cell, it can then engage in a binary manner with either the substrate or the E3 ligase. It can then go on to form this key ternary complex where the degrader molecule is now holding the E3 ligase and the substrate in close proximity. And it's this key structure which allows for recruitment of the E2 and ubiquitin, and then ubiquitination, polyubiquitination, and then degradation of this substrate protein. And so our aims are to understand what makes a good degrader, so understanding this pathway, which will then allow us to design and evaluate new degraders. And so as a case study for understanding what it takes for degradability with this mechanism, what we've done is we've decided to choose a model system based around the Protac MZ1, which was published in Alessio's lab back in 2015. And what we've done is we've inserted 7 changes on the substrate recruitment moiety, several changes in the linker region, and 12 changes in the E3 ligase recruitment moiety. And together we've assembled this Protac library of chemical analogs of MZ1, which allows us to consistently and systematically analyze single atom changes in the mechanism of action. And then we've then gone on to assemble a small catalog of biophysical assays that we can use to study this mechanism. So the first one is a cellular biophysical assay which was published by Rishing et al. 2018. And here we have a Brett signal between this nanoluke and a tracer molecule. And then upon treatment with the Protac, we then have a loss of Brett signal, which corresponds to engagement of the Protac with VHL within the cell. An example of a cellular degradation assay that we do is using full-length BRD4 CRISPR-Cas9 high bit knock-in. And then this causes luminescence of BRD4, which upon treatment with the Protac leads to loss of luminescence, which we can monitor in a kinetic manner. And then in terms of in vitro biophysical assays, we have this binary binding assay, which relies on the helix biosensor, where we have these DNA nanostructures where we can immobilize this protein. And then upon binding with the Protac, we have a quenching in this fluorescent dye, which we can monitor. And then for ternary complex formation, we're now immobilizing not one, but two proteins simultaneously. And then upon binding of the Protac, we have this closing of these arms, which allows proximity between this donor dye and this acceptor dye, which can lead to a FRET, so faster resonance energy transfer. So with this whole Protac library, we can map permeability, binary binding, ternary complex formation, and degradation. And there's a lot of results. But in order to just focus on a few key points for the purpose of this talk, let's think first about cellular uptake of the Protac, which is a key feature in degrader design. So we know that cellular uptake can be challenging with Protacs, and Protacs often violate Lipinski's rules. So they typically have much larger molecular weights than 500 grams per mole, and typically more than the hydrogen bond donor and acceptors are recommended. And this can often translate to lower in vivo drug absorption. So we know that cellular uptake is a requirement for degradability. And indeed, some of our experiments do demonstrate this. So here we have a small case study with these MZP Protacs. And you can see in this top left panel here that we have very good binary binding with VHL. However, when we look at this in the cellular context, you can see in the blue here that in the live cells, we have very poor intracellular availability and engagement with VHL. However, when we look at the ternary complex formation in vitro, we have quite good ternary complex formation. And then this then translates to a reasonably modest cellular degradation trace. So we know here that one of the limiting parameters for very good potent degradation is the cellular uptake and VHL engagement within the cell. And one of the things that we looked at was how to rescue this. So we actually saw that with other Protacs, this poor intracellular availability can be rescued by excellent ternary complex formation. So an example of this is with SIM1, which can recruit both bromodomains of BRD4 simultaneously. And what we see is that this Protac has identical binary binding to the MZP Protacs, but it has incredibly poor cellular VHL engagement. So it's barely entering the cell and engaging with VHL. But what we see is it has an incredibly stable ternary complex. And then this translates to really excellent cellular degradation, incredibly rapid and potent. So this is just one example of how ternary complex formation can rescue incredibly poor cellular uptake and availability. So if we now think about the second step in the pathway, which is binary binding, so binary engagement of the Protac with the substrate or the E3 ligase. So a quick example of this is the cis-MZ1, which features the cis-hydroxyproline epimer on this VHL binding moiety. And what this does, it abolishes any binding with VHL. So you can see that we're completely flatlining, which leads to a complete flatline in ternary complex and zero cellular degradation. So we know that binary binding with Protacs is required. It is a requirement for degradation. So what happens now if we modulate the affinity, so the binary binding affinity, and how does this influence degradation outcome? So we generated these two analogs of MZ1, which are fluorinated, and we have the two epimers of these in cis and trans. And what happens in the case of this Protac 88 is we have really poor VHL engagement, so very little binding, and this translates to quite modest degradation. In contrast with AT10, we're actually enhancing binary binding, so we have good VHL engagement, good permeability, and actually very good degradation too. So this is indicating that if we're abolishing some of the binary binding, then this is having a severe consequence in terms of our cellular degradation. But what we saw is with 88, we've actually got a 20-fold loss in binary binding affinity, which corresponded to a KD of 3 micromolar relative to MZ1. But what we noticed was that we actually only have a 2-fold loss in potency relative to MZ1 in terms of degradation. So this is actually yet another example of how one of these upstream steps, so in this case, binary binding, can be rescued to a certain extent by ternary complex formation. And the fact is that this Protac 88, despite having some reduction in binary binding, is still a very cooperative Protac. So the equilibrium of binding is pushed towards this ternary complex formation. And this is what is really driving the degradation. So ultimately, there's a lot of data and many more examples can be unpicked, but just really to quickly summarize for this section, in terms of degradability, what we're really thinking about is we're trying to deliver the Protac to its site of action. So it needs to be able to get into the cell, and we can optimize for that chemically. But what we also need is for the Protac to be effective at its site of action, and that means forming a stable ternary complex. And it's really the balance and cycle of design and optimization, balancing these two factors, which for us is driving degradation. So if we stop and think for a minute now about the right-hand side of this mechanism, one of the steps that we haven't talked about quite as much is ubiquitination. So ubiquitination has been, up until recently, a bit of a black box in targeted protein degradation field. And so if we think about the ternary complex, there's been a number of studies that show that enhanced ternary complex stability correlates with enhanced target degradation. There's also been a number of structural studies on the ternary complex. So a few examples here are with VHL, cereblon, and decaf15, where we've solved ternary structures with the Protacs in complex with their substrates. And we've done this with X-ray crystallography for VHL and cereblon, and also Cryo-EM for decaf16 more recently. And there are many more examples of these ternary structures. But one common feature is that all of these structures only focus on the substrate receptor and the adapter proteins and lack any information about the full assembly of the colon ring E3 ligase. And in that way, we're then lacking this information about ubiquitination. So what we wanted to do was see if we could solve this MZ1-induced ubiquitination complex. So we again turned to our Protac MZ1, which has a good KD and a reasonably long half-life, because we thought that this good degrader would be able to hold this ternary complex together for long enough for it to be vitrified and for it to be resistant to vitrification on a Cryo-EM grid. And then we assembled the other materials, so the substrate BRD4-BD2, and then the full E3 ligase, so the full Col2 RBX1 netylated complex with VHL, ELO-B, ELO-C. And we turned to cryo-electron microscopy to solve this structure. And this technique has been around since the 1930s. These are some of the initial pictures and sketches. Nowadays, the microscope relies on the same principles, but it looks a little bit different. And Cryo-EM has been a really exciting technique for us in drug discovery. It got awarded the Nobel Prize for Chemistry back in 2017. And one of the main driving factors for that is that historically, Cryo-EM used to be more suitable for very large protein complexes and was somewhat limited in resolution. But with the improvement in our computational infrastructure and also in our hardware of the microscopes, we've been able to achieve really amazing resolutions with Cryo-EM to the point where we can look at smaller proteins, more flexible proteins, and also even get volume for small molecules in these complexes. And so Cryo-EM works in a similar way to light microscopy. So, the principles are the same. We have a light source at the bottom and then a condenser lens, and then the specimen sits on this platform here. We then have an objective lens and a projector lens where we then can see the magnified image. And all of these are done with these glass lenses. And so in electron microscopy, we're kind of flipping this upside down where our electron source is at the start. And then we're using these electromagnetic lenses throughout the pathway to then see our magnified image at the bottom here. In reality, the system looks a bit more complicated. So you can see there's a lot of components that go into this. But the general principles really are that we're generating electrons in this first part of the column. We're then focusing the electrons, and then we're creating an image from the sample with the specimen here. We're then magnifying the image, and then we're capturing the image on a camera. And so all of this is done under vacuum and liquid nitrogen. The specimen support, so the Cryo-EM grid is 3 millimeters in diameter. So they're really very small and flimsy and can be tricky to deal with. And then the cameras that we're using are typically a thin sheet of silicon. And these are actually the same sheets that we get in our smartphones, although the ones in the electron microscopes are a lot larger. And so in order to create our complex for Cryo-EM study, we first had to express and purify the full-length E3 ligase, so the nedylated colon ring 2 VHL. And then we then incubated this with our Protac MZ1, the substrate BRD4BD2, and then also UBE2R1, which was stably loaded with ubiquitin. We observed full complex formation by mass photometry, observing a shift in mass. And then we went on to solve some Cryo-EM structures of the complex without the E2. And so what you can see here is that the Protac in yellow here is actually orienting the substrate towards the C-terminus of the E3 ligase for catalysis. And so we solved this structure in the University of Dundee with our Glacios operating at 200 kV. And then we went on to using a more powerful microscope at EBIC in Oxford, solve another structure with the E2 and ubiquitin bound, so the E2 in red and ubiquitin in yellow now. And what we could see with this structure was also a fair amount of conformational heterogeneity. So using some 3D variability analyses, we were able to capture movement and hinging of the colon in 2 different planes of motion. And so then when we built an atomic model into this structure, we saw that there were 3 lysines on BRD4BD2, which were in close proximity to the E2 catalytic site. So these 3 lysines highlighted in pink, in close proximity to the E2 catalytic site. So to look into this further, we performed some in vitro ubiquitination assays. And then my colleague Sarah in Ron Hayes' lab at the University of Dundee was able to excise the bands of the ubiquitinated substrate and perform mass spec of the ubiquitination sites. And in pink, she highlighted all of the lysine residues that showed evidence of ubiquitination in this in vitro assay. And then using our Cryo-EM structure, what we did was we were able to map these lysines onto 2 different faces of the substrate. So in pink, we have this face with ubiquitinated lysines, which we called the light face, which is actually oriented towards the C-terminus of the colon. And then in blue here, we have a cluster of lysine residues, which are on the opposing face. And these showed no evidence of being modified by being ubiquitinated in vitro. And so what we were able to do is kind of draw a line between the light phase and the dark phase of the substrate, where we were getting ubiquitination versus no ubiquitination. And so in order to verify the relevance of these lysines, we performed some lysine to arginine mutations. So we first generated the 8 times mutant. So all of these lysines on this light phase were mutated to arginines. And then we also generated this single mutant because we noticed that this lysine 456 was actually the only lysine that showed any modification after 30 minutes in the reaction. So we generated this single point mutant and also this triple point mutant with all 3 of these lysines, which seem to be in close proximity to the E2 catalytic sites. Then we expressed and purified these mutants and labeled them with an Alexa floor dye so that we could look at in-gel fluorescence and monitor ubiquitination. And we saw that with the wild type in green and the single point mutant in orange here, we had quite similar ubiquitination extent. And really this can be partially explained by how long-lived the complex with MZ1 is, which allows for the flexibility of the culling to buffer against the loss of this single lysine residue. But what you can see now is when we lose these 3 lysines with this triple point mutant, and also when we lose all 8 of these lysines with this 8-point mutant, we have a severe reduction in ubiquitination. So in order to look at whether this was relevant in cells, my colleague Connor in Alessio's lab transfected HEC293 cells with nanoluke BRD4 and looked at degradation on treatment with MZ1. And what he saw was when he treated with the 3-point and the 8-point mutants, we have quite significant reduction in degradation. My colleague Gajanan, also in Alessio's lab, was then able to perform global ubiquitinomics of this system. And then we were able to see which lysine showed evidence of ubiquitination in the cell. And what he saw was in pink, we have all of these lysines, which we previously mapped to our light phase. But he also found that there were 2 extra lysines which showed evidence of ubiquitination, which we previously mapped to the dark phase of the substrate. And so then when we looked at the structure a little bit more closely, we were able to see that these 2 lysines were in fact kind of on the cusp between the light phase and the dark phase. So really the longevity of this ternary complex and the flexibility of this culling were able to capture these lysines, which were kind of on the cusp and buffer against the loss also of all 8 of these mutants in the ubiquitination and degradation assays. So ultimately, what we've done is we've looked at the degrader MZ1, capturing the E3 ligase and the target BRD4-BD2. And then we've looked at how ubiquitin gets fired onto BRD4-BD2 to hit what we call this lysine bullseye. So this K456 residue, which appears to be, optimally positioned for ubiquitination by this system. However, what we've also seen is how the flexibility of the culling ring E3 ligase and the longevity of the ternary complex with the degrader can account for suboptimal lysines to also be captured. So ultimately, what we want to know is what makes a good degrader and how can we design better degraders? And, from what we've spoken about today, I think what we can say is that each step of the mechanism is important and that certain Protacs, such as MZ1, mediate this exquisite selectivity of orienting the substrate towards the catalytic E2 for ubiquitination and also orienting lysines for ubiquitination. And therefore, if we were to create more stable versions of this specific orientation of ternary complex, this could well be a very viable strategy to accelerate the addition of that first ubiquitin and therefore accelerate degradation. However, if we think about it, for more, let's say, suboptimal degrader ternary complexes, where let's say the degrader orientates the substrate away from the E2 catalytic site, we would then be relying on the longevity of this ternary complex and the flexibility of this cullen to capture these lysine residues, which would be deemed slightly less optimal for ubiquitination. But let's say we were to stabilize this ternary complex through generating chemical analogs. This could potentially lock this complex into an unfavorable conformation for ubiquitination. So ultimately, we're optimizing for cellular permeability and also for ternary complex selectivity. But an important factor to think about is also what that ternary complex looks like in relation to our ubiquitination complex. And so I'd like to finish there and thank my supervisors, so first and foremost, Alessio, and also Ron Hay and Hannah Maple from Biotechne, everyone who's involved in the Cryo-EM infrastructure, access and training across Dundee and across Scotland. We have some really excellent facilities and it's been really fantastic to obtain training over the past few years in Cryo-EM. The Chile lab, Biotechne, who sponsored my PhD, several members of the University of Dundee and CTPD, and my funders for my PhD, the MRC and Biotechne. And so thank you, and I'm happy to take any questions.